Hey everyone, I hope you can hear me. Yeah, it is. Okay, can you hear me? The question is, can you hear me? Um, la 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 la. Okay, I think I should be audible right now. Okay, let's do a check. Uh, so my name is Aishman. I am from India. Shit, yeah. Okay, so my name is Aishman. I'm from India. Can you hear me now? Yeah. Okay. So when the last time we were studying, my microphone went dead at this topic uh, on the membrane proteins. Okay. And today we will start from here, right? Right. So what I was telling you guys that there are proteins on the cell membrane. Okay. There are proteins on the cell membrane, and on the basis of age of extraction. Age of extraction means that how easily they can be removed from the membrane. They are divided into two types. Okay, first one being the integral, or we say, sorry, the first one being integral and the second one being peripheral. Okay, the peripheral one, they lie on the surface, while the integral proteins, they are partially or totally buried on the cell membrane. Okay, so here if you look, this one, it is a integral, it is totally buried in the cell membrane. This one, it is partially buried. Okay, so it is integral, and I don't think there's a another one, but like this, it is on the surface, it is peripheral. Okay, now on this basis, like, okay, I will tell you two things here. First thing, only integral proteins, only integral membranous proteins, what does they do? They makes the channels on the membrane, which helps in the transport of molecules from here to here. Okay, but the peripheral ones, they cannot do that. Understand? And now I am going to tell you about the type of transport through the membrane. So first one here will be the simple diffusion, the second one will be the facilitated transport, and the third one is active transport. Okay, so in the case of simple diffusion, what happens that a molecule or a substance, it moves from its its higher concentration to its lower concentration across the membrane. Okay, and in case of simple diffusion, it do not requires any sort of carrier proteins. Now let me tell you a bit about carrier proteins. When will we need carrier proteins? So As we know the structure of the plasma membrane, it has non-polar, sorry, it has polar head outwards and non-polar tail inwards. So the inner part, it is a non-polar region, right? So from the non-polar region, it is impossible for a polar particle to go across it. Okay, and for to pass a polar particle across the membrane, we requires integral proteins. Understand? And that's why when we talk about simple diffusion, and we say that it is across the concentration gradient and it do not required any integral protein. It means that the particle which will undergo the simple diffusion, they will be non-polar particles only. Okay, like water, it also move across the concentration gradient, but it is not a non-polar particle, so it will not do simple diffusion across the membrane. Okay, now we will talk about the facilitated transport. Facilitated transport. Okay, in this case. It occurs across the concentration gradient, just like the simple diffusion, but in this case, the polar molecules are transferred across the membrane. Okay, and if they are polar molecules, they require an integral protein, right? And when we talk about the active transport, it requires energy. Okay, like these two, they are the example of passive transport. But this is the active transport. Active transport do not requires active transport requires energy for its transport from one part to another part. Let me check the quality. I think it's good quality, right? It should be good. Just wait a minute. Just wait. Mm, Someone is watching. Oh, it's me watching from another US. Yeah. So now we will talk about active transport. Sorry, I was checking about the quality. Yeah, it's good, I think. So, in case of active transport, it occurs against the concentration gradient. Okay, and it also requires protein. So it doesn't matter the molecule is non-polar or polar. If it is the active transport, it is going to require integral proteins. Okay, because it is going to be against the concentration gradient, and if against the concentration gradient it will occur, it will also require energy, right? And if it will require energy, it will require a machinery which will help it to utilize that energy in transferring the particle against the concentration gradient as per requirement. Okay, and one thing that in simple diffusion, the rate the rate, it do not have any limits. Okay, but in case of facilitated transport and the active transport, there are limits. There are limits of rate. There are limits of rate. Now, when we talk about facilitated transport, so in this case, like it requires integral proteins. Okay, and there are limited number of integral proteins. Okay, so a when like a molecule is going from one integral protein from here to here, it has a capacity, right? That how much molecules can go per second, right? And when that capacity reaches, the rate cannot increase much further, right? And in case of active transport, there are also limits, and the reason of the limits will be first one being the integral protein, the limited count of integral protein, the capacity of integral protein, and the second one will be the energy, because active transport, it requires energy, and to transport molecules, if you will not have enough energy, the transport rate, it will be limited because of lack of required extra energy, right? um yeah. This part I'm going to skip it because I just told you about that there, and I will just tell you this. Okay, just wait a second. Yeah, I'm back. Mhm. Sorry. Hi. Well, so now we will talk about the fluid mosaic model. So, fluid mosaic model. So, like when we didn't know the structure of the cell, okay, like you know what, there are a lot of things in the science which are just based on theories. Okay, so when we talk about the cell plus cell walls, sorry, cell membrane structure, or we say plasma membrane structure, we are not always sure. We have multiple theories and we focuses on them and we try to prove them. Okay, so in the early 90s there are a lot of theories which came on the structure of a cell membrane, and the one which was most accurate was the fluid mosaic model. According to this, the quasi-fluid nature of lipids enables the lateral movement of proteins within the overall bilayer. So, what do we mean by quasi-fluid nature? So, these lipids, they have like this empty space as well, which is filled with sort of liquid. Also, like they are not tightly packed with each other. They keep moving from one place to another, from one place to another, and which helps the integral proteins to be also moved from one place to another. Okay, and there are two type of movements, two types of movement. The first one is flip flop. A flip flop. What do we mean by flip flop? In the case of flip flop, what happens like this phospholipid, it is here and it goes somewhere else and it goes like this. It replaces its position. It is called flip flop, and it only occurs in case of phospholipids. Okay, in case of integral proteins, it is not possible because they are very large and they have less mobility as compared to the lipids. Okay, and the second one will be lateral movement. Okay, in case of lateral movement, they can move in across the same membrane in which they are present in the same layer from here to there, from change their lateral location. Okay, and this can also be done by integral proteins. Okay, but it cannot be done by the what do we say, sorry, but integral proteins cannot do the flip flop movement, right? And this was all in this case. Yeah, also one thing, the fluid nature, it is a very important thing, that the fluid nature of the membrane, it is also important for from the point of view of function like cell growth, formation of intercellular junctions and secretions, endocytosis, cell division. So understand, if the nature of the, first of all, quasi fluid nature means having a liquid nature. Okay, not completely liquid, not completely solid, it has like semi fluid nature. Okay, quasi fluid nature is like semi fluid nature, and having this nature, it helps a lot. Okay, like if this is a cell, this is a cell, right? Let me do it. Okay, if this is a cell, it wants to divide, right? And if the cell wall is a solid, it will be impossible, I mean not impossible, but it will be very hard and very high energy consuming for it to divide. Okay, but if it has a semi fluid nature, it can divide easily without any sort of problems. Okay, it can stay intact when it needs to, like in the in its midlife, like when it is not going to divide, and it can easily take food by uh, keeping its other parts a bit more fragile, a bit more semi-fluid nature. Okay, it can manage itself and it can change regarding the conditions. Okay, and it helps in like cell growth. A cell can easily expand. More phospholipids can join it. More proteins can join it easily, and it can easily expand, which is not possible in a solid case. It can easily form intercellular structures, junctions. It can easily do secretion, endocytosis, right? Now we are going to talk about the cell wall. Okay, so as you may recall, a non-living rigid structure called the cell wall, it forms an outer covering for the plasma membrane of fungi and plants. Um, so when we talk about animals, we do not have a cell wall, which I told you in earlier lectures as well. Animals do not have cell wall, but if you look at plant cells, if you look at fungi, they have cell wall. What is a cell wall? Cell wall is a extra layer around the plasma membrane of a cell. Its function are generally just being rigid and protecting cell from certain chemicals or macromolecules. Okay, and a cell wall, it gives shape to the cell. It also protects it from mechanical damage and infection. Okay, it also helps in cell to cell interaction and provides barrier to undesirable micromolecules. Okay, one more thing, that the cell wall is not alive, it is sort of dead. It do not interacts, it do not controls like what molecules will come to the cell or not. Now you will say that it is contradictory, that you just said that it controls and you are saying it don't. So how does cell protects how the cell wall protects the cell from undesirable macromolecules is in the word here, macromolecules. Okay, macromolecules means that they have large size. So cell wall, it do not have so large pores. It do not have large pores. It has smaller pores. And if it has smaller pores, it is impossible for the things with the big size to come inside the cell, right? And algae, it also have cell wall. Algae cell wall is made of cellulose, keratin, mannans, and minerals like calcium carbonate, which I don't think it is a important information for us. While in plants, cell wall is form of cellulose, hemicellulose, pectins, and proteins. The cell wall of a young plant cell has a primary wall. Okay, so what happens like when a plant is young? Let me do a good one. Yeah. So let me do it again. Yeah. When it is young, a young cell. Oh, sorry. Sorry. I'm sorry. I'm sorry. Okay. When it is young, so what happens that the new cell wall, it is called primary cell wall. What are its feature? It has ability to do what? It has ability to grow. Okay, it can grow in size, and what happens that slowly with life it diminishes, and a new secondary cell wall forms in between the primary cell wall and the cell wall. Okay, here is the new cell wall, which is known as secondary cell wall. Okay, also there is a presence of a thing called the middle lamella. which is mainly made of calcium pectate, which holds or glues together neighboring cells together. So this is the cell wall of one cell, and if there is cell wall of a adjacent cell, then there will be a layer. This layer, which is called middle lamella, which helps in gluing two adjacent cells together. Okay. And one more thing, that cell wall and middle lamella, they both may be crossed or intersected by the plasmodesmata. Okay, plasmodesmata. What does plasmodesmata do? It helps in transferring material or cellular material from one cell to another. Okay. Now we are going to talk about endomembrane system, and believe me, it is a very interesting part, a very interesting topic of this subject or of this chapter. Okay, let's take a little 2 minute break, and I'm telling you again. Okay, just 20 seconds. Okay, let's start now. Mm, We are going to study about endomembrane system. Okay, so let's start now. We are going to study about endomembranous system. Um, as I told you earlier, there are compartmentalization in the cell of a eukaryotic cell. Okay, like like this, like this, like here. Okay, and what do I mean by compartmentalization? That is different function now divided into different organelles. Okay, they are divided by different. They are done by different organelles, different function. Okay, and we are going to study about endomembranous system. So, while each of the membranous organelle is distinct in terms of its own structure and function, many of these are considered together as endomembranous system. Okay, and why are they together considered as an endomembranous system? Because their functions, they are coordinated. Okay, the function of one depends on the function of another. The function of another depends on the function of another than that. Okay, the endomembranous system, it includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Since the function of mitochondria, chloroplast, and peroxisome are not coordinated with the above components, these are not considered as the part of endomembranous system. What do we mean by that? So, in endomembranous system, what we are considering is endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. What we are not considering? Mitochondria, chloroplast, and peroxisome. Why? Why we are considering them, but we are not considering them? Because the basis of endomembranous system, the organizing and endomembranous system, is that the components in the EMS, their functioning is depends upon each other. So here the things which endoplasmic reticulum makes. It goes to Golgi complex, and from Golgi complex becomes the lysosome. From lysosome to vacuole. Okay, their functioning it depends upon each other. But in case of mitochondria, chloroplast, and peroxisome, they do not depend so upon either of them. They work independently. Okay. Now we are going to study about them one by one. The first one being the endoplasmic reticulum. Okay. So. Um, the electron microscope studies of eukaryotic cell revealed that the presence of a network or reticulum of tiny tubular structure scattered in the cytoplasm. That is called the endoplasmic reticulum. So, when there were microscopic studies done on the eukaryotic cell, what scientists found were like tiny tubular structure, like this tiny tubular structure, okay, into the cytoplasm of a eukaryotic cell, and they called them the the endoplasmic reticulum, okay. Hence, endoplasmic reticulum it divides the intracellular space into two distinct compartments, that is, luminal inside the endoplasmic reticulum and extraluminal cytoplasm. So, uh, when you look at the endoplasmic reticulum, you look at this area, like it is present in the endoplasmic reticulum, okay. And when you look at the outside area, it is present outside the endoplasmic reticulum. And on the basis of this, there are two types. There are two types of space around the endoplasmic reticulum. First one being the luminal inside endoplasmic reticulum. Second one being the extraluminal, um, which are the cytoplasmic compartments, okay, or we say outside the endoplasmic reticulum. The the endoplasmic reticulum it often shows ribosomes attached to their outer surface. Okay, and when there are ribosomes attached to the outer surface, it will be called a rough endoplasmic reticulum. Okay, and if there are absence of ribosome on the surface, then it will be called smooth endoplasmic reticulum. Rough endoplasmic reticulum is frequently observed in the cell, actively involved in protein synthesis and secretion. They are extensive, continuous with the outer membrane of the nucleus. So, rough endoplasmic reticulum is frequently observed in a cell with continue with the outer layer of the nucleus. Okay, and the smooth endoplasmic reticulum is major site of synthesis of lipid. In animal cells, in animal cell, lipid like hormones like steroidal hormones, they are synthesized in the smooth endoplasmic reticulum. Okay, do you understand? I hope you do. Okay. Now we are going to study about Golgi apparatus. Um, and for today, we are going to finish up to the vacuole, right? We will finish the endomembrane system. So, Golgi apparatus. It is a good topic. Yeah, I like it. Golgi apparatus. It was discovered by Camille Golgi, first observed in 1898 by the help of densely strained reticular structure, it was observed as a densely strained reticular structure near the nucleus. Okay, there were later named Golgi bodies after Camille Golgi. They consist of many flat disc-shaped sacs or cisternae, which has like zero point 0.5 micrometer to 1 micrometer diameter. These are stacked parallel to each other, they are parallel to each other, but they are in disc-shaped, sac-shaped like this. Okay. There are a varied number of cisternae that are present in the Golgi complex. The Golgi cisternae they are concentrically arranged near the nucleus with distinct convex cis-oval forming face. So, mm, there are two types of phases in the in the Golgi complex. The first one being a near phase. Near phase. What do I mean by near phase? That when you look at the endoplasmic reticulum here, let me zoom a bit little. Okay, just wait. Okay, when you look here on the endoplasmic reticulum, like here, so um, there are no good. Mm, okay, I think I can. Yeah, yeah. So this here. So when you look at the okay, I mean, I'm having some issues. Okay, I will tell you on my own. Just do it. Okay. So there are two phases of the Golgi apparatus. Okay, so like if we think that this is the endoplasmic reticulum, the first phase will be a convex phase. So like look at a single cistern. So it is the endoplasmic reticulum and it is the Golgi apparatus. Okay. If there's a face near the or towards the what do we call it endoplasmic reticulum, it has a convex shape and it will be called forming face or it will be called cis face. And when it's the and on that cisternae the opposite side which is to opposite to the endoplasmic reticulum, it will be called transface or sorry, I think I forgot. It will be called transface or maturing phase. Yeah, or maturing phase. Okay. The cis and trans phases of the organelle they are entirely different, but they are interconnected. This side of the Golgi apparatus and this side they are totally different, but they are interconnected. Okay. The Golgi apparatus principally performs the function of packaging materials. The materials which goes from endoplasmic reticulum to the Golgi apparatus, they are being packed. Okay? And to be delivered either to the intracellular targets or they are secreted outside the cell. Materials to be packed in the form of vesicle from the endoplasmic reticulum, they fuse with the cis phase of the Golgi apparatus and then move towards the maturing phase. So what happens that whenever endoplasmic reticulum releases something, it goes and fuses to the cis phase and it goes out from the trans phase. Fuse to cis phase goes out to the trans phase and this process happens and it is being packed in the form of vesicles. This explains why Golgi apparatus remains in close association with the endoplasmic reticulum. So it is a very common thing that if they are working together and the Golgi apparatus is packing the things which are releasing from the endoplasmic reticulum, then endoplasmic reticulum and Golgi apparatus, what do they need? They need to be very close to each other. Okay. A number of proteins synthesized by the ribosome on the endoplasmic reticulum, they are modified in the cisternae of the Golgi apparatus before they are released from its transphase. So, what happens like when there are proteins which are synthesized on the ribosomes of the endoplasmic reticulum, they are transferred to the Golgi apparatus and they are modified there. They are being modified there, and then they are released from its transphase. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. In Golgi apparatus, like what happens that proteins which are sent from the endoplasmic reticulum, they are transformed into glycoproteins, and lipids which are sent from the smooth endoplasmic reticulum, they are converted into glycoproteins. Okay. Now we will study about the lysosomes. So they are membrane-bound vesicular structure. They are formed by the packaging of Golgi apparatus as they are related. Okay, and they are in a term like endoplasmic reticulum, then Golgi apparatus, and then lysosome. The isolated lysosomal vesicle they have been found to be very rich in almost all type of hydrolytic enzymes. Okay, and what are these hydrolytic enzymes like hydrolase, lipases, proteases, carbohydrases? Okay, carbohydrases, and they are optically active at the acidic pH. Okay, and if they are active at the acidic pH, what is the nature inside the lysosome? It is going to be alkaline pH because they are not active in a normal cell. In a normal case, they are being activated only when like a cell wants to destroy itself or cell wants to destroy some particular substance. Then either lysosome will be sent to it or the particles will be sent to the lysosome for the destruction. Okay, these enzymes they are capable of digesting carbohydrate, protein, lipids, and nucleic acids. Now we will study about vacuoles. Vacuole is the membrane-bound space found in the cytoplasm. It contains water sap, excretory product, and other materials not useful for the cell. So, vacuoles are a single membrane-bound structure found in both in eukaryotic cell, and they contains generally extra product or waste product in it. Okay, like extra water, extra sap, or like excretory product, which is a waste product, or other products like maybe ions, which are not useful to the cell at that particular moment, and which can be used later. Okay. The vacuole is bound by a single membrane, which is called tonoplast. In plants, the cell vacuole can occupy up to 90% of the volume of the cell. But in case of animals, it is very, very different. Okay, in plants there are generally only single vacuoles, but in case of animal cell, there could be multiple small size vacuoles. In plants, the tonoplast facilitate the transport of a number of ions, other materials against the concentration gradient into the vacuole, against the concentration gradient. So, there are what are present here integral proteins, or we say carrier proteins, or we say um, transport proteins. Okay, which helps in the transport or which by the active transport process against the concentration gradient in the vacuole inside the vacuole from the cytoplasm. Okay, hence their concentration is significantly higher in the vacuole than the common than the cytoplasm. In amoeba, the contractile vacuole is important for the osmoregulation excretion. In many cells, as in protista, food vacuoles are formed by engulfing the food particles. Okay, so I mean in amoeba there are contractile vacuole. Contractile vacuole have ability to contract, and which helps it in its motility. Okay, and its food vacuole are formed by engulfing the food particles. Like I think we all know about that. What happens like if this is the cell surface of the amoeba and there's a food particle outside, then slowly what it does that it will engulf the food particle like slowly. Okay, we all know about that, right? And I think that's it for our today's lecture. I will meet you tomorrow. Bye bye everyone, take care, stay together.